Well, good morning, and thank you very much for the opportunity to speak to you all today. It's a real privilege to be here. The work that I'm going to describe was primarily done during a 3-year fellowship paid for by the Nuffield Department of Population Health at the University of Oxford. And as was said, I'm presently a postdoc working on the Compute project, which I will be doing for the next 2 years or so. But as was said, my background was as a physicist, and for those of you who know physicists tend to sort of think of the world in terms of sort of simple models that allow you to imagine what might happen and to make calculations. And the multistage, so when I joined the Cancer Epidemiology Unit in 2016, I found myself sort of searching around, trying to find a sort of conceptual picture that helped me to sort of understand how cancer may arise and how that might influence the age distribution of the instance of it. So it didn't take very long before I came across the multistage model of carcinogenesis. So what is it? Well, as was mentioned, the basic idea is that cancer may arise through, say, a sequence of genetic mutations. You can have a few of these mutations and you'll remain perfectly healthy. It's not until that final stage has occurred and you've triggered the cancer that you've got a problem. And what the authors realized was that if each of these stages, be they, for example, mutations, were to occur at an approximately constant rate, then this would lead to a very particular distribution for your risk of disease as a function of age. And if you were to plot your data in an appropriate way, then you should see a nice straight line, and the slope of that line would tell you something about the number of these steps needed to trigger the cancer. And so this is a figure from the paper, and this, so you see, yes, nice straight lines, all looks pretty good. And they must have been very excited by this at the time. But as you go through to the 60s, the 70s, the 80s, and the 90s, the model doesn't really occur very often. There are reasons for this. So even today, many people find it difficult to sort of formulate and understand the mathematics of the model. There may have been issues fitting more general versions of it to the data. There are questions about the identifiability. Can you really uniquely determine the parameters and be certain that that's the number of steps that are involved? But perhaps more importantly, alternative methods came along, so logistic regression, proportional hazards, that were perhaps more suited to many of the questions that were being asked, questions to do with relative risks. So if you smoked, how much does your risk of one disease increase relative to somebody who doesn't? But as George Box famously once said, all models are wrong, at least with enough data, they tend to be wrong. I mean, this is why particle physicists build bigger and bigger experiments to find smaller and smaller things that are wrong so they can move forward the field. But some are useful. And the multistage model of carcinogenesis is certainly one that has proven to be very useful indeed. So when it has appeared during the past few decades, it's usually been associated with important pieces of work to do with retinoblastoma or colorectal cancer or myeloid leukemia. But it was really around about sort of 2015 or so that there was a real explosion of papers related to this topic. And this arose from the availability of genetic sequencing of cancers. So you could really start to interrogate what was going on in reality. And for me, perhaps the most important sort of thing to come out of this sort of new wave of work was the recognition of just how common precancerous clones are as we age. So if you look at blood in an old person, it's not unusual to find that a proportion of the cells consist of what are genetically different clones that have mutations and they're coexisting with your original blood cells in a perfectly sort of harmless way, as far as we can tell. They can be and some of them can be precancerous, but they're generally harmless. And everywhere that people have tended to look so far, similar things have been found. So if you look at the tissues in the throat, in the skin, I mean, the skin is a lovely example. I mean, many of us will have moles on our skin. As we get older, we typically get more of them. They often change. If you're a trained doctor can look at those and say, well, those changes are benign, or whether there is a problem and you need to have a local anesthetic and a 20-minute procedure to get them removed. So in a sense, this shouldn't really have come as a surprise to us, but nonetheless, it's this work is really sort of clarified the situation. So an obvious thought is, well, if we're sort of increasingly, if our body is increasingly consisting of these clonal populations of cells which have mutated as we age, could they be involved in other diseases? And by coincidence, during this similar sort of time period, there was some other pieces of work on amyotrophic lateral sclerosis in particular, or ALS for short, which I believe is known as motor neuron disease. Also some work on Alzheimer's and Parkinson's disease. And typically what these studies would do was very much like the very first paper by Armitage and Dole. And so you'll get a plot, an appropriate sort of plot of the data, and you see this nice straight line, and that would indicate that the model was consistent with, so the multistage model was consistent with the instance of disease. So in this case, this is Parkinson's disease. It's just one of the diseases in our study. It's just there by chance, so I was checking that the data was all fine. And the different colored curves are for different ethnicities. But there's another reason why you might expect a multi-stage model to be able to describe a lot of different disease types. And that's because in its most general form, a multi-stage model is any model where you go from a healthy to a disease state through one or more independent pathways through either a sequential set of steps or a non-sequential set. And whenever that's the case, then you can write down a mathematical equation for the age distribution. And so that's what I show in that paper there. However, in practice, there will typically be one pathway by which disease occurs most rapidly. And that's what will dominate your observational data, and that's what you'll see. And when that's the case, then for most diseases in most people, then you can approximate the model with what is, to the statisticians in the audience, a Weibull distribution. So the HR is the cumulative hazard function, E to the beta X is the relative risk, T is the age, L is just a scaling factor, and K is the parameter which is related to the number of steps in the model. So how common are these diseases? Well, when I was doing this work, I happened to be working on UK Biobank data, and I was looking at 400 diseases in men, 400 of the most common diseases in women, and I fit the data to as many of those as possible. And I found about 60% of them were consistent with the model. It would probably be less than that if there was more data, but it is a substantial proportion of the diseases that I was able to look at. I've just got some random examples picked there. So you might start to wonder, well, why hasn't this been noticed before? And there are several good reasons for this. One is simply that the data would not have been easily available. But another reason is the mathematical issue of left truncation. So just to explain what I mean by this. So if you imagine you are born and you consider your chance of surviving without getting a disease. So initially when you're born, that's 1. But as you get older, that probability of surviving that disease falls. Now, most data in most cohort studies is from people between a certain age range. So UK Biobank, for example, recruited people between ages, say, 40 to 60. So by that time people are being recruited into the study, that survival curve has already dropped below 1. Okay, now if you have data over the entire lifetime and you plot it appropriately to test for a multi-stage model, what you would see is this lovely straight line, and you'd be assuming, yes, that's great, that's consistent with the multi-stage disease process. But if your data is only collected, say, from age 40, you'll see something very different entirely. And you might well conclude, quite reasonably, but incorrectly, that it's not a multi-stage disease process. However, you can make a small correction. It's very simple to do, actually. I'll show it to you in this paper here. And when you do that, then you regain this nice straight line. Now, this is simulated data. That's why the lines are all so clear. Okay, so well, the next question is, okay, well, you've got another statistical model. We've got plenty already. Why bother? But as George Box says, again, so, some models are useful, and model is particularly useful for providing insights. And I'm just going to give 4 examples of the sort of new perspectives that this way of thinking can provide. So, having looked at hundreds of diseases in UK Biobank, one of the most obvious things that came away was how the age dependence risk varied between these different disease types. Now, if you use logistic regression or proportional hazards, you're just really getting a relative risk. You're not seeing the age dependence of disease. But when you look across hundreds of diseases, what becomes very clear is that whereas some diseases are more sort of sporadic, so they have a kind of a low risk throughout your lifetime that increases only slowly, other diseases, the late onset diseases, they have a negligible risk throughout most of your life. But then as you get older, they rapidly increase in probability. So if your interest was in trying to reduce the burden on the NHS, you'd probably be wanting to target those more sporadic diseases because they're the ones that look most amenable to reducing the likelihood of. And I'll say more about that in a moment. Another useful concept that emerges is effective age. So what do I mean by that? Well, the Weidel model is a particular type of proportional hazards model. So if you take the relative risk there, e to the beta x, and you put it inside the brackets, so the k now needs to come down over here, then what you're left with, what that whole term represents, is your effective age in terms of your risk of disease. So if your relative risk is bigger than 1, then your effective age is larger than your actual age. And if it's less than 1, then it's less than your actual age. And your disease risk is equivalent to somebody with no risk factors, but your effective age. Now, why would this be useful? Well, when you're screening people for disease, you don't want to overdiagnose or underdiagnose people. So if you have a lot of risk factors, then you probably want to screen them sooner or more frequently. But if you have few risk factors, you probably want to screen them less because you don't want to have unnecessary procedures being made. But if you grouped people and screened them based on their effective age, then you would be screening people who are at an equivalent risk of that disease. This term, which has been a modified version of the relative risk, is now like an aging rate. And so the sporadic diseases which have a low value of this parameter k, your aging rate is much greater. So your risk of a sporadic disease is much more modifiable than for these late onset diseases where this parameter k is higher, which suppresses the effect of the term, the size of the term. Another thing you can do is you can take a strong risk factor, be that genetic or an exposure, such as smoking, which is the example I'm going to show, and you can use that to stratify your data, so to split your data into 2 separate groups. And then if you plot your data in an appropriate way, like as before, you can then interpret differences between the data using the multi-stage model. So, so the smoking data is in red, the non-smoking data is in blue. And so if we go across to this side, first of all, we can see this sort of rigid vertical displacement of the two curves, which in terms of the model would be interpreted that there's just a general increase in the rate of all the processes. So if it were just mutations involved, it would be a general increase in the rate of mutations. In the middle plot, the curve has gone up and the slope is reduced. Now that's exactly what you'd expect to see if you've increased the rate of one of these steps sufficiently that it's no longer a rate limiting step. So there's fewer steps in the disease process. This one for lung cancer on the right is a little bit unusual because the curve has gone up and it's become steeper. Does anybody have any thoughts as to what might be going on in that particular case? Well, my best guess on this is that the lung cancer that you get in smokers is typically different to the type of lung cancer you get in non-smokers. So although you will have a few cases of that, the type of lung cancer you see in smokers and the non-smokers, it'll be dominated by a different type of data. When you smoke, it increases your risk of this other type of cancer. So what you're effectively seeing there is you're effectively seeing a different disease, and that's why you're seeing the different slope. At least that's my interpretation. The final example I'm going to mention is multimorbidity. And the reason why I've got an excuse to mention this is because this paper appeared a month or so ago. It makes some rather strong claims about the importance of multi-stage models for modeling multimorbidity, but it does give me an excuse to talk about the topic. So multimorbidity is the presence of two or more diseases within an individual. And the difficulty we have with studying or treating multimorbidity is that once you get one disease, it can modify your risk of another, either through the disease itself or through treatments or changes in your behavior. And we really just cannot model that sort of situation at the moment. So a year or two ago, what I thought I would do was I'd tackle the one problem we can do in a fairly rigorous way, which is to consider disease risk in people whom are otherwise healthy. So the idea was to model the disease risk in otherwise healthy people and predict in the UK Biobank cohort how many disease we'd expect in people who had no prior disease. That's the points in blue. And there's a lovely straight line because it's just confirming that the model is working. I then compared that with what we actually observed over that period, which are the points in black. And there's two sort of fairly surprising things about this. I mean, one is the increase is fairly modest. It's about 50%, which actually is not that large compared to how other factors such as smoking may increase your risk of some disease types. And the other thing is that this increase is surprisingly similar across a whole range of different diseases. So one possible explanation for this is that if there were shared pathways influencing multiple disease types, then we might expect different groups of diseases to be increasing by a similar amount. And this is further work that needs to be done. Which brings me on to my present project that I'm working on. So I'm in the statistics department, working on the compute project as part of a sort of a group of other people across Oxford and the UK. And just very briefly, so the problem is that multimorbidity is very common amongst people in the UK, and it's becoming more so. We don't know how to treat people who have many things wrong with them or even to study them. And what it will require is a much greater sort of overall general understanding of diseases and of how they interact with one another. Now, the picture that I have, and I think is emerging and being adopted more broadly, is that you've got a situation, we've got, say, aging and exposures on the one hand, acting through some sort of shared mechanisms or pathways leading to clusters or patterns of diseases. So on one hand, you might have studies looking to identify the clusters of diseases, and then the hope is that we can somehow combine all this work to identify the pathways, so you may target them with a drug or some other treatment in future. So just to finish off, I'd like to turn to some words by Professor Armitage's co-author, Professor Richard Dole, who said that death in old age is inevitable, but death before old age is not. And based on what I can see here, I think I sort of certainly agree with the first part, that death in old age is inevitable, and hopefully also death before old age is also avoidable. But I think for this century we need a bigger ambition, and perhaps ill health is not inevitable. So perhaps for this century, for the challenge we should have is for the medical community and for public health is to identify how we can avoid ill health and ideally to broaden, make use of that to establish a healthy population going forward in the future. But one thing that I am sure about is that the multi-stage model of carcinogenesis will be talked about and discussed for decades to come. So I'll just like to thank again my funders, the Nuffield Department of Population Health and NIHR. And if you have any more questions, please do come and talk to me about them. Thank you for your attention.